Okay, thanks. Okay, well, the few of you who've arrived, thank you for coming. Please feel free to study your pathology notes. I'll just talk to myself up here, and I will have Sean cut little pieces out of the tape so the people who didn't come will be misled when it comes to the exam. So you all have an advantage. I thought it was particularly apt when we walked in here, we're talking about geriatrics, care, etc., that Dr. Mayer should walk in and be, of course, a prime example of that. Either when the hair goes grey or at least disappears completely. End of life care. We're approaching it. Okay, but hopefully it won't be initiated by viral disease. And what we're going to talk about this morning is, in this hour, is going to be antiviral drugs. And antiviral drugs are something that are very important, but on the other hand, we have another way of attacking most viral diseases, which of course is via vaccines, or many viral diseases. So if we're looking to develop a drug against a virus, then we have to think about the same sort of things that we have to think about in regards to bacteria. And with bacteria, we have a lot of antibiotics that will kill off bacteria. And of course, they've been extremely successful. We started off with the penicillins, and we've gone through a large number of other antibiotics, and they still, to a great extent, work. And the reason, of course, they work and they don't damage us is because of the high selectivity of the antibiotics that take advantage of differences between prokaryotic and eukaryotic cells and attack the bacterium by attacking something which is the bacterium's Achilles heel. That is something that is unique to the bacterium, no effect on us because we don't carry out our metabolism in the same way. And of course, the trouble with antiviral chemotherapy has always been that viruses are parasites on our cells. And in most respects, they parasitize the metabolic pathways that we carry out to reproduce viruses, to get more viruses. And so it's very difficult to get selectivity because you can, when you start to interfere with the metabolism of the virus, in most cases you're going to interfere with the metabolism of the host cell. And of course, one thing to remember is that the most effective way you can treat your patients who've got a viral disease is to kill the patient, because that will very effectively also kill off the virus replication and the viral disease. However, hopefully you will not be doing that. If you do decide to take that approach, please let me know so that I don't actually come to your clinic. But antiviral chemotherapy raises the whole problem of toxicity of the drugs to the cell and to the host organism. So the key is selectivity, finding that virus's Achilles heel, and you're going to have not only properties with toxicity, you're going to have the properties that you have with all drugs, and that is rapid excretion. The patient doesn't cooperate. You give the patient the drug, and it promptly excretes it. Rapid metabolism, so that something that is active in the test tube against the virus or in the cell culture dish turns out to be completely inactive in the patient because the patient unfortunately metabolized it to an inactive form. And of course you've got to get the drug into the patient, and poor absorption can be a problem. So the ideal drug is going to be something that's water-soluble, you can get into the bloodstream, it's going to be reasonably metabolically and chemically stable once it's in the bloodstream, it's going to be able to get across the plasma membrane, because in this case, unlike with the many bacterial infections, we're looking at an organism that is inside the cell, so you're going to have to get it into the cell. That means it's probably this drug is not going to be charged. It's going to have to be something that's apolar that will go across the lipid bilayer of the plasma membrane. And of course, you don't want it to have any of these properties of carcinogenesis, allergenic, mutagenic, teratogenic, etc. So you're going to have to do a lot of work on a drug that you're developing for clinical use. And what drug companies like to look at is a thing called the therapeutic index. And this is simply the minimum dose that is toxic to the cell over the minimum dose that's toxic to the virus. And you want to have in an effective drug at least 1,000, a TI of at least 100 to 1,000. And some of the better drugs that we've developed in the past decade or so have TIs much better than that. And that means they are really very selective. And of course, when you're developing drugs and you're worrying about carcinogenesis, you're worrying about toxicity, and you're going to prescribe some of these kinds of drugs which are available, which have these nasty effects, then you've got to take into account the problem that your patient is suffering from. Obviously, if they've got a runny nose, a rhinovirus infection or something, you're not going to give them something that's carcinogenic. On the other hand, if you've got a patient coming in with symptomatic rabies, then you might want to think of some more aggressive treatment. Although for symptomatic rabies, you probably aren't going to find any drug that's going to be of much use. So, considering that vaccines have been so effective, why do we even bother with drugs? Well, there's good reasons for that. Many established diseases don't have a suitable vaccine when the disease is caused by a virus. Retroviruses, as we talked about over the past 2 hours, have got a high degree of mutation, and every vaccine that has been developed so far against things like HIV, the virus mutates and escapes from the effects of the vaccine. Problems with the current vaccine. There's always a problem with the current vaccine for influenza. It has to be redeveloped every year, as you know, and sometimes, although they do a pretty good job of predicting the circulating strains in which the current year's vaccine is against, nevertheless, there have been failures in the sense that they have not predicted the strain that will circulate during the winter. And, of course, new and emerging diseases, hantaviruses, which I think you'll probably be hearing about in the next hour, is a good example where a disease emerges that has been not seen very much before, and as a result, there are, of course, no vaccine. Vaccines take decades to develop. That's why we're still struggling with HIV vaccines, but even other vaccines are going to take a long, long time to develop, even when the virus is more amenable to antibody neutralization. And, of course, when you've got patients who are immunosuppressed, vaccines are going to have problems there. So at present, no drug completely suppresses viral replication, with the possible, and I think you can now say with the probable exception of the anti-HIV protease inhibitors used concurrently in heart therapy with 2 other drugs, where there is now emerging good evidence that the viral replication is stopped. But as I told you yesterday, there's still this problem of viruses taking cover in memory cells. So if you're going to develop your vaccine, you're going to have to look for a specific viral function. And, of course, there are specific viral functions and enzymes that are necessary for the virus life cycle that the virus provides for itself. Or you're going to have to look for a cellular function that the virus needs in order to replicate. But if you're going to interfere with a cellular function, then you have to interfere with a function that's crucial to the virus, but is not crucial to the cell. Or, if you are going to interfere with something that's crucial to the cell, then you're going to have to target that drug. You're going to have to target that drug to virus-infected cells. Get rid of them, but you don't kill off all the other cells in the body. And that's how one of the better drugs that we have these days works. So what can you target in terms of viral-specific functions? Well, of course, you've heard over and over again in these lectures on viruses that many viruses code for their nucleic acid synthesizing, genome synthesizing proteins, their enzymes, in the form of DNA-dependent DNA polymerases, RNA-dependent RNA polymerases, and in the case of the retroviruses with the reverse transcriptase, the RNA-dependent DNA polymerase. So these are things which the virus produces, along with proteases in the retroviruses, that integrase that I told you about yesterday, and, of course, in the orthomyxos, the flu viruses, neuraminidase. These are all virus-encoded proteins that are essential to the lifestyle of the virus, and if you get rid of them, then you have a good chance of selectively, and particularly important, selectively knocking out the virus. And as you can see here, the history of chemotherapy is littered with failures, but there have been a few successes. The very first approved antiviral drug is up here. It's idoxuridine, and you can immediately see what it does. It's clearly a pyrimidine analog. It interferes with virus DNA replication. It's however toxic, but it has still found use as a topical drug. One of the better drugs we have is acyclovir here, a really good anti-herpes drug, very widely used. Again, you can see what it does. It's a purine analog. It's modified here in the sugar, and by being modified here in the sugar, it can't form a phosphodiester bond because there's no hydroxyl group to participate in phosphodiester bond formation, and as a result, it's a chain terminator, and we'll come back to it again in a moment. And in the 1990s, we've got the protease inhibitors. So there are a large number of stages of the life cycle in which we could at least think about inhibiting the virus. We could think about inhibiting the binding of the virus to the surface of the cell. That's clearly important and often very selective. We could think about stopping fusion, either here at the cell surface in the case of the herpes viruses or HIV, or if endocytosis and the low pH of an endosome, maybe we could stop the endosome getting acidic and stopping the virus getting in there. That's going to be rather non-specific, though, for other cells. But if the virus does get into the cytoplasm, it's then usually in the form of a nuclear capsid that has to be uncoated. Maybe we could attack that, because that is something that's rather specific, that uncoating of the viral genome in order that it can be expressed. Then we've got possibly integration in the case of the DNA viruses or retroviruses. Maybe we could attack that. RNA export, maybe that's a possibility. This part here is going to be a big problem because it's translation of the viral proteins, which is virtually the same as translation of our proteins. And so it's not surprising there are no good antivirals that actually act in this part of the life cycle. But out here, assembly of the virus, then that might be a possibility. If we can stop the virus maturing to an infectious form, then that's also a possibility for developing an antiviral agent. So let's just run through some of these stages of the life cycle and see what's being done. Now, binding to the cell surface receptor, not really much to brag about there. People tried with HIV soluble CD4. If you could compete with the CD4 on the cell surface with soluble CD4, then maybe it could act to stop the binding of the virus to the CD4 antigen on the surface of the T4 cells. People worried about that because they thought, well, maybe if you put in a soluble CD4 to block the GP120 binding to the CD4 antigen on the cell, then since we know co-receptors are involved here, maybe you could actually exacerbate the problem by providing the CD4 and still having the virus bind onto the co-receptor, the chemokine receptor in the cell. That turned out not to be a problem, and some companies developed this CD4 derivative. It's a recombinant protein, and it seems that it can reduce virus levels in vivo. It's a recombinant protein which contains a part of its soluble is CD4 antigen, and then it's also linked onto immunoglobulin G. So this drug, CD4IG2, is available. It's been tried in the clinics. It can reduce the levels of virus, but it's not the answer to an HIV infection. A couple others of chemical drugs have been developed against the fusion, process, although this one here, AMD 1300, actually binds to fusin, which is one of those chemokine receptors. And there's also this drug here, which RFI 641, which is active against respiratory syncytial virus fusion. They're not widely used. These have got into the clinic. These are peptides. These are peptides that look like important parts of the GP41, which is that other part of the surface glycoprotein of HIV, and which is involved in the fusion of the membrane of the virus to the plasma membrane of the cell, and entry of the nucleocapsid into the cytoplasm. And so if you can bind on this peptide here to the proteins on the cell surface, then you can inhibit the fusion after the virus is bound. And T20 has achieved quite a lot of success in terms of reducing levels of virus in patients in the clinic. And then because of the success of that, a new generation, which is actually a different peptide, but still a peptide from GP41, has now been developed, and that is undergoing trials as well. However, again, on their own, they're not the answer to an HIV infection. But when patients develop resistance to the heart therapy, then these are possibilities that can be developed to achieve another attack on the virus that may be useful. Now we go to the fusion of the membranes and entry into the lysosomes. There are drugs which were originally thought to alter lysosome metabolism, and these include the anti-influenza amantadine and rimantadine. And this one, amantadine, was developed in the 1960s. Rimantadine came along in the 1990s. They're only effective, as you've probably heard from the flu lecture, against influenza A. They do reduce flu prophylactically, and they haven't been used much here because of the good vaccines. They were widely used and effectively used as a prophylaxis in the countries of the former Soviet Union. And of course now with the idea or the problem coming up of bird flu, where we have no suitable vaccine, then people are reinvestigating some of these drugs in order to use them chemotherapeutically in possible bird flu outbreaks. they've been used in this country primarily for people who don't get a good response to a vaccine. And they tend to be elderly people, immunocompromised people, people who are allergic to eggs in which the vaccine is produced, and where you find a flu strain coming up which is not in the vaccine. Originally, these were thought to be lysosomotropic drugs. Now we know actually they're not. They affect the M2 protein of the virus, and this alters the maturation of the virus via some effects in the Golgi body, maturation of the virus surface proteins, meaning that the virus that comes out of the cells tends to be less infective because the surface glycoproteins haven't matured properly. Another way of interfering with viruses would be to stop uncoating, as I mentioned. Here's a drug that was used originally to do just that. It's called araldone, and it seems to act against picornaviruses by stabilizing the coat by some unknown mechanism, not widely used. Here's one that has been used successfully in some circumstances. It stabilizes also the coat of picornaviruses. It's one of the Win drugs. We know how this works. This is the active site here of the drug. And there's a similar derivative of this called pleconaril, which you may have read about. This is a derivative of the Win drugs. It's really nicely absorbed by the patient, gets into the brain. And unfortunately, the old formulation, which you took orally, didn't work too well because of the way the patient metabolized it. But there's a new formulation out from Schering-Plough now, which is a nasal spray that seems to have at least some effectivity against picornaviruses. And the way it works is by entering the VP1 protein here. Remember, there's a canyon in the VP1 protein. The canyon alters its conformation when the virus is uncoated and allows the RNA to come out of the virus. If you can stick something that stabilizes the conformation of this canyon in the VP1 protein, the RNA doesn't come out, and the RNA, therefore, the genome remains encapsulated in the nuclear capsid, and therefore the virus is non-infective. But most of our drugs have been against nucleic acid synthesis. Often, as we've said, the polymerase that duplicates the genome is encoded in the virus. But it's not only that. In some viruses, the very large viruses, such as herpes viruses, there are other enzymes that are encoded in the genome of the virus. And that includes the thymidine kinase of herpes virus. So herpes virus not only encodes a polymerase that allows it to copy its DNA into more DNA particles and thereby duplicate the virus, it also, in the cell, sets up an environment for good copying of DNA by providing the constituents for DNA polymerization by encoding also a thymidine kinase. And this is what it does. Here is thymidine, or deoxythymidine, in fact, and this gets phosphorylated. The first phosphate group is put on by usually the virus here, in the case of a virus-infected cell, and then these two other phosphates come on from cellular kinases. But here you are making deoxythymidine triphosphate. Now, in cells that are resting, such as nerve cells, then you don't have much of the constituents of DNA, because why would the cell be producing the constituents of DNA when it doesn't want to replicate its DNA? The presence of the thymidine kinase allows the cell to make the constituents to polymerize into DNA, which is what the virus wants it to do. So why does it code for its own thymidine kinase? Well, we know that TK- viruses, these are the ones which have had the thymidine kinase knocked out, they can't grow on non-proliferating cells because non-proliferating cells don't make DNA. So even though these cells, like nerve cells, for example, they have the purines and pyrimidines, the level of the monophosphorylated nucleosides are usually very, very low. And so the virus resolves that problem to grow in things like nerve cells by phosphorylating the nucleosides that are present around in the cell. Actually, thymidine kinase is a bit of a misnomer, because you and I do this job of phosphorylating our deoxynucleosides with 4 kinases, one for each of the nucleosides that are going to be put into DNA. The virus presumably can't afford to take up too much of its genome doing this, and so it has evolved what is really a deoxynucleoside non-specific kinase, though since it was first assayed in cells, it's usually referred to as the thymidine kinase, which will phosphorylate, monophosphorylate all 4 of the nucleosides to give monophosphorylated nucleosides. And so this is a rather non-specific enzyme here. And as I said, the herpes thymidine kinase will phosphorylate any of the deoxynucleosides. Because it is so non-specific, which is inevitable because there's only this one enzyme to do what the cell uses 4 enzymes to do, it's very non-specific, and therefore, fortunately for us, it'll not only phosphorylate the real nucleosides, it'll also phosphorylate chemicals that look like nucleosides, some of the drugs that we're going to use to attack the virus in the cell. So this nucleoside analog can be given as a non-phosphorylated form, which is great, because it doesn't have that phosphate charge on it, so it gets across the plasma membrane. And then the thymidine kinase inside the cell will add a phosphate to it, and then the drug will become activated, and it will be used by the host cell DNA duplication system and get incorporated into DNA and then interfere with DNA metabolism. So this is nice and selective because it's only the cells that contain herpes virus which will actually phosphorylate the drug, because only the cells which have herpes virus have this non-specific thymidine kinase that can be acted upon by that can act upon the drug. Now, certain herpes viruses, cytomegalovirus is an example, Epstein-Barr virus is another example. They don't have their own thymidine kinase, but what they do is to kick the cell into producing a hell of a lot of its own thymidine kinases and other nucleoside kinases. And although it says here the cellular enzymes don't phosphorylate the drug, if there's really a large amount of those enzymes, they will phosphorylate the drug to a certain extent, and you'll get enough phosphorylated drug to do the job. But there's still this selectivity because it's only the virus-infected cells that have got this large amount of the enzymes that can activate the drug. So therefore, the need for activation restricts the drug to viruses such as HSV that code for their own enzyme, or viruses such as cytomegalo and Epstein-Barr that cause the cell to overproduce. But again, to emphasize, it is the selectivity that's important here, because only the cell that's infected by the virus activates the drug. So the thymidine kinase provided by the virus activates the drug, but then the target of the drug is the DNA polymerase, the DNA duplication system. And we've got a variety of these drugs. There are some which are sugar modifications, and there are some that are base modifications. Also, it turns out that the polymerases of the virus are often more sensitive to these drugs than are the cellular DNA polymerases. So that's another observation in their favor, because it makes the drug even more selective. So you get the drug selectively activated, and then when you've got an activated drug, it actually targets better the viral enzyme than it targets the host cell enzyme. And here's a couple of examples here. These are really pretty good drugs. Acyclovir, we mentioned, came up in the 1980s. Ganciclovir followed it. They're very, very similar, and you can obviously see how they work. They have changes or they are modified sugars here so that they can't participate in phosphodiester bond formation. They get activated by the viral thymidine kinase. They are too different from the normal nucleoside to be an effective substrate for your cellular deoxynucleoside kinase. This is an excellent herpes drug. You've probably seen it advertised widely on the television. There was a nice ad some years ago where two young people walking down the beach hand in hand into the sunset discussed the effectiveness of acyclovir and how it had changed their life because of its effect on their genital herpes. So a widely used drug. Ganciclovir has found more of a niche for itself in Epstein-Barr cytomegalovirus infection. And again, to emphasize how these work, here's DNA being formed in normal DNA synthesis, by base pairing, adding on the correct nucleosides here, and so you get an extension of the chain. With the case of acyclovir, what happens is once acyclovir gets base paired here, then there's no hydroxyl group for the virus, for the DNA polymerase to continue on polymerizing the DNA strand here. So, very effective against a variety of herpes viruses. It can be taken topically, it can be put into the patient intravenously. Unfortunately, like all of the drugs we have against viruses, resistant mutants arise, but it's not teratogenic, it's not carcinogenic. And as I mentioned before, ganciclovir has found a nice niche against cytomegalovirus. It's used, for example, in cytomegalovirus-induced retinitis in HIV patients. Here's another one of the earlier ones, adenine arabinoside. It's a competitive inhibitor of the DNA polymerases, which is more sensitive to this drug than the host cell. However, just looking at these problems here, it's not a drug that you'd want to put in large amounts into one of your patients. But it's found a niche also for itself as a topical application for herpes simplex. It also has other problems. It's rather unstable. This amino group here is metabolized off by the patient to form an inactive form of the drug. So deamination is a problem, but it can be used topically against various herpes virus infections. Here are a few others that act against DNA polymerases or interfere with DNA metabolism. You've all heard of AZT here, azetothymidine. Here's DDI, dideoxyinosane. And you can see how all of these work. They lack the hydroxyl group that is necessary here on the sugar, the ribose, to carry on chain elongation. Some of them are not sugar modification. Here's one, Veroptic, and here's idoxuridine again, which have substitutions on the base here, on the case of both of these on pyrimidines, and they just simply alter the base pairing, so you just induce lots and lots of mutations in the virus as it's duplicating itself, and you get non-infectious, non-functional virus because there's just such a huge mutation load. But again, you get resistant mutants. And here's a guy which has two substitutions, a fluorine here on the sugar and also an iodine here on the base. This is fluoroiodoaracytosine. Here's an interesting one, famciclovir. Famciclovir is given to the patient as a prodrug, and Dave Gangemi might have mentioned this to you. Famciclovir here is an ineffective compound, but it can easily be taken up by the patient's cells. It's non-polar. It gets into the cells. It's metabolized to penciclovir, and then that penciclovir can be phosphorylated, first of all, by the HSV, herpes simplex virus, thymidine kinase, and then the cell puts on two more phosphate groups, and that can now interfere with cellular DNA metabolism. This penciclovir, which you can also apply that as a topical cream, but if you're going to have it orally, you give famciclovir, it comes from GSK, and then it gets metabolized over to penciclovir by the host. So that's some of the drugs that are involved in directly interfering at the active site of the polymerases of various viruses. And when HIV became a major problem, AZT was the first drug. It acts on the reverse transcriptase. It's a reasonable drug to give. It was the only drug to give at the very beginning here. But the problem was the patient soon developed resistance to the drug, didn't have any effect anymore. And so people thought, well, maybe we should use two drugs. One drug that would attack at the active site would be a competitive inhibitor side of a drug attacking the active site of the reverse transcriptase, and maybe we could take another drug and that would attack the somewhere else, bind to somewhere else on the reverse transcriptase. And so a resistant mutant at the active site, which would make it resistant to AZT, would not necessarily be a resistant mutant that would stop another kind of drug binding to the reverse transcriptase. And so the non-nucleoside, non-competitive reverse transcriptase inhibitors were developed. And these work in combination with AZT or similar drugs to AZT like DDI. They are very potent non-competitive reverse transcriptase inhibitors. They work in the nanomolar range, and they have virtually no toxicity at all. You don't normally need a reverse transcriptase. In fact, you don't need reverse transcriptase to carry out your metabolic processes. These are very, very specific inhibitors of that one enzyme, and as a result, they have minimal toxicity. They're very good bioavailability. They're absolutely useless in most cases in monotherapy, but they are very good in combination with other drugs. And they are one part of the heart therapy that we use against HIV today. Here's some structures here, and I should state, while I remember to tell you here, I don't remember the precise structures of any of these drugs, and I don't expect you to either. I mean, clearly, you're intelligent people. I assume you can recognize something that looks like a pyrimidine versus a purine. But you don't need to memorize the structures of these kind of things. Nevirapine was one of them, approved for AIDS patients, good blocker of mother-to-child transmission. This is one of the cases where monotherapy does seem to work. Now, a single dose given to mum when she comes into labor will reduce HIV transmission by 50%. Give a dose to the baby within another 72 hours, and you get an even better effect. And nevirapine is quite widely used to suppress mother-to-baby transmission of HIV, and particularly important in places like Africa, where there's been so much transmission, either in utero or alternatively by breastfeeding. This drug has shown great efficacy in doing that, and in fact is made available by the manufacturer of the drug free of charge in third world countries. So it shows you that at least some drug companies have a bit of a conscience. So nevirapine, a very useful drug in stopping or at least reducing mother-to-baby transmission of HIV. Another of these is Sustiva, and that's frequently used as a combination of heart, as is nevirapine. Both of these are approved for use in HIV patients. An older drug here, phosphonoacetic acid and phosphonoformic acid, has found some use. It also binds to the polymerases. It's a competitive inhibitor. It tends to be more selective for the viral polymerases. It's extremely toxic, and you get rapid resistance. But it may be useful against cytomegalovirus, has gone into a clinical trial. Now, this guy here is a bit different. You've heard about this one, again, I think from Dave Gangemi, ribavirin. Ribavirin, you immediately have noticed, I'm sure, it's got two hydroxyl groups here, so it's not a DNA base, a DNA constituent analog, it's an RNA constituent analog. And it's found uses in various places, such as respiratory syncytial virus. You can puff it into the patient, and you can also against some of these really horrible things like Lassa fever, Korean hemorrhagic fever, and those kind of things, where your patient is really in a pretty bad way. It's found some use there. We don't know exactly how it works. Some people suggest that it works by interfering with cap formation on RNAs. Remember, there's this methylguanosine cap on messenger RNAs. Then it's thought that maybe the ribavirin interferes with the metabolism of this, but it's also thought it may induce just random mutations in RNA viruses, because it actually has quite a bit of efficacy, at least in the test tube in the culture dish, against poliovirus, which doesn't have a capped messenger RNA. There was a deal of enthusiasm some years ago for ribozymes. These are catalytic RNAs that can very specifically cleave certain sequences, and a drug was developed called Heptazyme. It's a ribosome that specifically cleaves hepatitis C RNA at very highly conserved regions, and so in the test tube, again, it looked rather good. When it got into the clinic, it was rather disappointing. And there used to be a very nice website on Heptazyme with lots of movies on how this worked, but unfortunately, the website, along with the company that was making it, disappeared. No inhibitors against viruses that go against protein synthesis, for the very good reason you act against viral protein synthesis, you act against the host cell protein synthesis. Protein processing, however, has been, in the past couple of decades, the site where there has been a lot of investigations and a lot of success. Glycosylation, there has been a drug called castanospermine that comes from the An Australian chestnut tree found some use in clinical trials, but it seems to have been dropped now. But proteolysis, the specific proteolysis that I emphasized to you, is so important in the retroviruses because they make these large polyproteins, and before the virus can be properly assembled and matured into an infectious particle, these proteases have to chop up the polyprotein. And if you don't chop it up, the virus can, to some extent, assemble, but it can't reinfect another patient. And what happens, of course, here is you've got this long polyprotein. In fact, both the GAG and the POL are made as one long polyprotein in the retrovirus. So it contains the GAG, the group-specific antigens, the integrase, the polymerase, and the protease at the end. The protease cuts itself free. It's an autoprotease activity at this stage. The protease cuts itself free, and then it binds back to the polyprotein and cuts it to form the polymerase, and then it cuts off the integrase. One of the earlier of the protease inhibitors was sequinavir, and these are aspartate proteases. The enzyme is an aspartate protease. I mean, it cuts at aspartate groups, and this is a derivative of a dipeptide here that fools the enzyme, binds to the active site of the protease, and inhibits it. And here is an example when you use another of these, indinavir, and you can see how a few weeks after using indinavir in combination therapy with two of the competitive inhibitors of reverse transcriptase AZT and 3TC, how it just brings the level of viruses down to almost at least, or in fact to undetectable levels. So it's in combination here in heart, it's extremely effective. And there's a whole series of these. They always end up usually with the name Inavir at the end, indinavir, nelfinavir, ritonavir, sequinavir, from different companies, but they all work in the same way. They're all inhibitors of, site-directed inhibitors of the active site of the aspartate protease that's involved in maturing HIV. And in the early stages, sequinavir in heart was shown to reduce viremia, CD4 cells went up, AIDS deaths were found to go down, and now these are one of the drugs of choice. So heart consists either of two nucleoside inhibitors plus a non-nucleoside inhibitor like nevirapine or Sostiva, or the two nucleoside inhibitors plus a protease inhibitor. And these it's sort of a choice as to which starts first, but if a patient does develop resistance to one heart regimen, then you can try another. As I said, with these things like indinavir in heart, no detectable HIV by PCR, which is, and in fact probably no detectable replication because we can't see any of the polymorphism developing, and so you can get it down to a very low level, no copying, no polymorphism. Therefore, you don't escape from the drug regimen by developing resistant mutants. So no replication means there's no resistance, and patients have been on this now for a good few years, and they seem to do very well. However, originally, these drugs were difficult to take because although it's called combination therapy, you don't take them all at the same time. And so some need to be taken on a empty stomach, some need to be taken with food. And so it was quite a complex ritual that the patient has to go through with 3 different drugs. Now the FDA has approved a combination drug which actually has one pill, which has several drugs mixed together. One of the problems was, of course, these drugs are made by different companies. Getting them together to form one drug, one pill, was somewhat difficult. They produced it. In fact, companies abroad produced it, but getting it available in this country was very, very difficult indeed. But if the patient stays on this, no resistance, and things seem to people do very well. Unfortunately, because of this rather complex regimen, people did come off it, the virus came up again, and then produced resistant mutants. There are side effects. One of these is a thing called buffalo hump, and it's a lipodystrophy, where you've got abnormal fat deposition in patients, and there also are other side effects. Some people find these difficult to tolerate, some people find them very easy to tolerate, exactly what you would expect in sort of broad population. But there's no doubt these have been exceedingly useful drugs in combating HIV. Finally, and we're getting around to the end of our hour together, influenza. You've been hearing all about bird flu and that kind of stuff. And there were a couple of drugs developed in the latter part of the last decade. And that people thought, well, why on earth do you want to develop this kind of drug? Because we have a really effective vaccine. Well, and they really didn't find much use except in patients who needed chemical prophylaxis, such as people who didn't respond to the vaccine in a bad way, or people who didn't respond to the vaccine because they were immunosuppressed. And these are the neuraminidase inhibitors. Now, flu uses neuraminidase containing proteins to latch onto cells. And these, it needs the neuraminidase, first of all, to escape from the cell once it's been made. It doesn't want to bind back onto its own receptor. So it uses the neuraminidase to cleave the sialic acid from the receptors on the surface of the cell it's just infected so that it can now drift off away from the cell that has produced it. And also, the virus has to get through all the muck in your lungs to infect other cells, and that mucus contains lots of glycoproteins, and those glycoproteins contain sialic acid. And the virus doesn't want to stick to the muck, it wants to get to the cell where it can do its replication, and so it uses the neuraminidase also to penetrate the mucus. So in 1997, Relenza came on the market, and this is a neuraminidase inhibitor. And one of the nice things about it is that Relenza is active both against A and B influenza strains, whereas the other ones, the amantadines and rimantadine, is only effective against influenza A. And here's the structure of them for your education or for your interest, but it's something I don't expect you to remember. So the Relenza was the first one. Relenza has to be taken as a nasal aerosol type of thing. Tamiflu, however, came up after that, and Tamiflu can be taken orally. So here is the way they work. After neuraminidase inhibition, the flu virus will retain the sialic acid on its surface, and therefore it binds back to the cell that's infected, or it will just bind to the to the hemagglutinin rather on other virus particles. So they will just clump together and they'll be non-infectious, or it'll just stick to the mucus in the respiratory tract. Here's the virus that's producing the cell that's producing the virus. The neuraminidase stops these sialic acids here being cleaved off, and as a result, the virus tends to stick onto the cell that it's just come out of. So these seem to be effective in some ways. As I said, nasal spray for zanamivir, Relenza, it shortens the symptoms by a few days. It's probably not going to affect most people. It's not going to be much use in most people. If you've got the flu, you might as well just go through it for a few extra days. On the other hand, when you add up the number of days lost to flu in the winter flu season, maybe that is of economic significance. But given to people who've already got the flu, it may just mean lesser symptoms. However, it's quite good, I think, prophylactically. So zanamivir, which I think was Relenza, wasn't it? Well, I don't remember now. Relenza is zanamivir. And that's approved for people over 7 years of age. This one is Tamiflu, or seltamivir, is approved for younger people. Prophylaxis can be used for. It's unfortunately, with the amantadines, as I said, they have a rapid resistance that emerges because of the mutation of the M2 protein of the influenza virus. With the case of these neuraminidase inhibitors, zanamivir and oseltamivir, there is resistance that develops, but these resistant mutants don't seem to spread around very much. But the data for some of these effectiveness things are rather limited at the moment. However, in people who have gone through some of these clinical trials, there are now 10 clinical trials that the CDC has sponsored, that it looks like the risk of pneumonia amongst the people who are participating in these trials was about half. So they are drugs that affect the consequences of flu. They're not so wonderful as treating the patients who've already got flu, but they have some effectiveness, at least in the as a prophylactic drug. In the case of bird flu, they're more or less all we've got. Some of the strains of bird flu that have emerged in Southeast Asia are resistant to amantadine and rimantadine, largely because of the use of these drugs in veterinary use. Feeding large amounts of them to poultry, of course, was never a good idea to stop them getting various bird diseases. And so since bird flu, obviously, they pass through poultry, then resistant mutants have arisen in the birds in Southeast Asia and presumably, therefore, will possibly spread to human beings. So far, there's not too much evidence that the bird flu is resistant to the neuraminidase inhibitors any more than the regular flu is. Okay, well, thank you all for coming. So good luck with the PATH exam. And so if you want to hear more about viruses, you'll get somebody who knows far more than I do about viruses, who will come along and talk to you about... Oh, she's standing up there making faces. He'll come along and talk to you